Welcome, everyone, to Gems from the Wisdom Traditions, a conversation circle that Stella Moving Center sponsors on Saturday afternoons. And this particular topic is very in keeping with the topic for the entire year of evolving with the cosmos. And the topic is evolving the human body. So there's a certain agency that is implied, I think, with that topic, evolving the human body. Um, I'm going to read a quote from Maria Popova for Monday, since there's a quote for every day that corresponds with our theme for the week. That every single atom in your body, if tagged and traced back in time, would lead to the core of a particular star in the early universe is a truth pulsating with transcendence. Maria Popova. And then one that comes from actually the esoteric Christian tradition. It's from Jesus for today, which is Easter. Excuse me, tomorrow, which will be Easter. If the flesh came into being for the sake of the spirit, that is a mystery. But if the spirit came into being for the sake of the body, that is a wondrous miracle. So some place between those two, between this more spiritual perspective and this more scientific perspective, I think is this idea of evolving the human body. And one of the most exciting aspects of today's science to me is called epigenetics. And we have with us Kelly Fraser, who actually has genetics as her career. So I'm going to let her tell you a little bit more of the specifics of that. She has been a wonderful contribution to Stella Moving Center ever since she moved in across the street from us and has been attending many of our different activities and has become more and more involved with Stella Moving Center. It's really a pure delight for me to introduce Kelly Fraser to all of you.
Thank you very much, Renee. And I'm going to have to tell everyone that I have given many talks in my time, but it's typically for scientists. So this was a very interesting perspective for me to try to figure out how to take all that science and translate it into something for non-scientist. And in doing so, I actually learned quite a bit. So it was great. And I'm hoping that I can convey my enthusiasm and what I actually learned by doing this. So
Wonderful.
the title of my talk is Epigenetics, How Your Environment Talks to Your Genes.
Oh, have I started record? Yes, I have. Go ahead.
Okay.
Yeah.
So this is just a teeny bit about me. I was born and raised, I don't know, do you see my pointer?
Yes.
Okay, I was born in, I wasn't born, but I was raised from a itty bitty age in Sunnyvale. This is the San Francisco Bay Area. And from there, I went to UC Santa Cruz, where I double majored. I got a BS in chemistry and a BS in biology. And from there, I moved up the coast, not too far, to UC San Francisco, where I got my PhD in genetics. And then across the bridge to UC Berkeley, where I did my postdoctoral work. And then after this journey, I thought I should get a job. And so I went and I worked in this little circle here. This is the stretch, sometimes called referred to as Silicon Valley. And I worked in a biotech company for 7 years. And after the 100th person told me I was too academic, I said, okay. So I left and I went down south to San Diego and I got a job in the UC San Diego Medical School as a professor and doing biomedical research. And now I am slowly bouncing back and forth as I transition into Honolulu. And I made this map this morning, and that's exactly where I was sitting. So you can see I'm very close to the Still and Moving Center. So I have a couple slides that are coming up that are some science, and then I go into some just casual things, and then I go back into a little bit of science. So, but first we're going to define some of the words that we're going to be using just to make sure that everybody's on the same page. So genetics, what is genetics? Well, this is the study of DNA, the DNA sequence. It's our DNA sequence is fixed. It's inherited. We don't change it. It's in every single cell. It controls the function of every cell in our body. And so then what is epigenetics? Well, if you think about it, you know, our stomach cells and our brain cells and our skin cells, they don't look the same and they don't function the same. But you just told me that they all have the same DNA sequence. So epigenetics is the way that our DNA is expressed. It is the mechanism by which our DNA sequence is controlled. It opens up DNA to express genes, sometimes large amounts of that gene, sometimes just a teeny weeny bit of that gene, or it will shut the genome down and say that gene's not going to be expressed anymore. So it's the epigenetics is why our cells look so different from each other. All right. And it's, you know, as we age, our epigenome changes. Our DNA sequence never changes, but our epigenome changes. So when we say that the epigenome, the epigenetics is the way that the environment talks to your sequence, what we're referring to is that we can modify our epigenome by some of the choices that we make, our lifestyle choices. So this is one of those science slides. What's genetics? What's genomics? So the genome, genetics is the study of genes, and genomics is the study of the genome. And the genome is in total these 23 autosomes, chromosomes, as well as the 2 sex chromosomes, the X and the Y. And it's really, there's 4 nucleic acids, adenine, thymine, cytosine, guanine. And it's really these 4 nucleic acids that encode our entire DNA. There's about 3 billion of them. And it's the string of 3 billion sets of these 4 nucleic acids distributed across our 22 chromosomes that compose our genome. And this is what we inherit from our parents. It is what makes us who we are. Okay. And the epigenetics and the epigenome, there's a slightly less people probably have heard about this. But so let's look at this panel C here. See the black? The black is representing DNA. It's representing that ATGC. And the blue ball is representing what's known as histones. So you have 4 different proteins come together and form a ball, and the DNA wraps around the ball. Okay? And so it's really the histones getting the DNA organized. And then when you modify those histones, okay, it's sometimes referred to as epigenetic marks or histone modifications. And we're going to focus here, we're going to go through this one right here. This is histone 3, all right? And we have different amino acids make up a protein. And so if we have histone 3, this the 27th lysine in that histone, K27. Okay, this is a common amino acids that is frequently modified, and it's modified by either adding a histone, so it's known as H3K27 acetylation. And what that does is it says open up. So that causes genes to be expressed. And then if instead of putting a histone on that lysine, we put a methyl base, a methyl moiety, it shuts it down. So that gene is not expressed. So this is what's referred to as the epigenome or epigenetics. And again, across all the cells of our body, we have the same DNA. Every cell has a vastly different epigenome. Not every cell type. All right. So when identical twins are born, right, they look very similar to one another. It's hard to tell them apart. And this is because they have the same DNA. And they've largely been in the same environment. Not exactly, but largely, right? And they look identical. When they get older, these are two identical twins and they dressed identically. But ironically, when I saw this, I just took this off the internet. I thought, they really aren't identical. If you told me they were sisters, I would say yes, but they're not identical. They don't look exactly the same, even though they're trying to, right? And this is because their epigenomes are not identical. Our epigenomes change through our life, and they're impacted by, what we eat, how do we exercise, do we sit in the sun? All of these things impact our epigenome. And I also ran across a very interesting series of identical twins. And this was put out by a Chinese photographer, Gao Ronggyo. I had never seen this before, but when I was thinking about how to put this talk together, I came across this. I thought, this is really interesting. And he says, he did have some females, but for whatever reason, I just picked 3 men, used to have the same face, living in the same family, but their lives changed due to various reasons after growing up. And so I don't think he's actually thinking of the epigenome, but that's exactly what's going on here. Now, this last pair on the right, the one on the left actually had an accident when he was 10. And so I don't think identical twins typically don't have that vast difference in height, but that was probably in part contributed to that. But again, these adults are not identical. Their facial structures, their skin tones, their skin makeup is not identical to one another. So, it's hard to, I'm going to try to have an interaction, but I can actually really only see 3 people on this screen. So I'll ask, what epigenetic marks, histone modifications, what do you think we can do? What behaviors and personal choices do you think we can make to affect our epigenetic marks? Does anyone have any idea?
Exercise.
No? Exercise.
Diet, emotions.
Diet, emotions, exactly. Everything that they tell us is good for us. It's good for us because it's affecting our epigenome, right?
I imagine whether or not we smoke.
Oh yeah, that's a real big one, right?
Or what we smoke.
or what we smoke. Yep. How often we drink, right? All of this is really, really impacting our epigenome. And so now I got to figure out how to, my screen sharing paused. Hold on. Now I was able to figure out how to see everybody. Now I can't figure out how to go back. Hold on. I don't want to leave.
Share.
I go back to share. Is that what I do?
Yes.
Okay, share settings. Well, hold on, let me see if I do that. Share. Oh, here we go. My desktop. I don't really want to share my desktop. Hold on.
What you're doing right now works pretty well.
Oh, okay. I got it. Okay. So there's all types of things we can do, right? Our sun exposure, right? We make vitamin D by interacting with the sun. And vitamin D is actually, people call it a vitamin, but it's actually more of a hormone. So when the sun hits your skin, right, certain rays, the UVBs penetrate through, and it interacts with 7-hydroxycholesterol, so it's cholesterol, and it causes a vitamin D precursor, which is very labile, and that forms vitamin D3, right? And this is very interesting because they say that over 50% of people in the United States are vitamin D deficient. And our lifestyles have really changed. We sit inside a lot. When we go outside, we tend to lather ourselves all up with sunscreen. And so, there's ways to supplement it. I'm a proponent of supplementation, but when I was putting this talk together, I listened to somebody who's an actual expert in this particular mechanism here. And it's, in his opinion, he's thinking that perhaps that the sun is actually generating, that the byproducts of this reaction may be actually generating more than vitamin D3. So his investigation is, what else could it potentially be generating that we don't know? So, I mean, I think, I don't have the answer, but how do we regulate our sun exposure? Our food. I mean, I picked vitamin C, but there's vitamin E, there's vitamin B12, there's all types of vitamins. And what do we eat? Do we eat a balanced meal? Vitamin C is important because it works through this TET enzyme, which promotes DNA demethylation. And if you remembered, we talk about how methylation closes things. And so without vitamin C, your DNA is not being regulated correctly. And that's true with all the vitamins in different ways. Exercise. Somebody said exercise. When you exercise in your muscles, your fat, your cardiac tissues, the DNA, histone, and the histone modifications are occurring. And these modifications in one tissue results in the expression of particular target genes in those tissues, which then we release metabolites and other things that react with the rest of the body. So when they say that our actions are really affecting our epigenome, this is exactly what they're talking about. Now, this one, I had something on stress, and then I didn't like what I had. So I just put this in just a teeny weeny bit ago. And this is relationship of stress. I mean, stress can cause huge changes in the way that your brain cells are working. And sometimes it's hard to undo those stress-induced changes. And one particular thing is post-traumatic stress disorder. So this is a disorder that everyone, I'm sure, has heard about, but what happens is it's not just some emotional thing. It's that your DNA, your epigenome has been modified due to this stressful disorder. You can imagine that you have a very stressful event, and your mind, your body wants to somehow keep that memory so that you don't do it again.
Mm.
So it changes the way that your mind is, your epigenome is regulating your cells. Okay, so then how can you actually undo that? Well, it's very difficult. We don't really have things that we can't order epigenome modifications. We can go through these activities that we do. But one of the things people are trying is this mindfulness-based stress reduction. So it's a meditation approach that I don't really know much about, but they say that it's effective in helping people with post-traumatic stress disorder. And so when you look at the biology underneath it, what's happening? Well, the methylation of some key genes is being brought back into a normal state, particularly probably the serotonin one here. So these activities that we do, even meditation, is changing our epigenome. So, and this I added because of a conversation I had with Renee earlier this week. So this is slightly different than our epigenome and our genome, but it's a topic that Renee was very interested in. So I thought, okay, I'll slip it in here. And that is when we think about our body and the cells in our body. In like 2018, they began to put out these statements and the realizations. So 8 years ago, that what we look at when we look at our body, our body is only 43% of the cells in our body are human. The rest are microscopic colonist. And we call these the microbiome. And they're in our gut, they're in our mouth, they're everywhere. They're on our skin, and they live with us. And, they have evolved with us and we have evolved with them. And so the taking care of our microbiome is almost as important as taking care of our body. And as we know, when we take antibiotics, which, a life-saving medicine, thank God for antibiotics, but we not only kill the bad bacteria, we kill the good bacteria. So we have to think about how do we take care of our microbiome? And it's very similar to how we take care of our own DNA. And that is that exercise and eating well is really, really important for gut health. And so these are quotes. I don't know this individual, this Dr. Sarampudi, saying that making dietary changes, you can change the composition of your microbiome quickly in days, eating yogurt, eating sauerkraut, miso, kimchi, whatever is fermented, whatever has good microbiome. I remember a few years ago, I was reading, and they think in the last 50 years or so, the microbiomes in humans, the complexity of the microbiome has really, really gone down a lot. And you can imagine that the microbiome isn't just a parasite. The microbiome helps us digest our food. The microbiome helps us release vitamins. So by decreasing the complexity of the microbiome, it's really hindering us. So it's very important that to you know, when to eat the foods which will help our microbiome keep healthy. So now I'm going to move a little bit into a more sciencey part for a minute. And this was a symposium I attended in Kyoto, Japan, a few months back. And we were all celebrating. You can see that's me. We were standing next to each other, and neither of us quite figured out how to move over here. But we were celebrating Dr. Yamanaka, Shinya Yamanaka, 20 years of his Nobel-winning discovery. And it was a major discovery that has impacted science tremendously. And that discovery, to put in simple words, was that you could take adult cells, you could take skin cells from an adult individual, and you could alter the epigenome of those skin cells, and you could bring them back to an embryonic state. So I clearly remember when learning about this, because we had been taught for decades. I mean, it wasn't a theory, it wasn't a hypothesis, it was the truth. That that biological development was a one-way road. You could never come back. So once you made a skin cell, that was it. Once you made a stomach cell, that was it. You could never reverse the process. But what Shinya Yamanaka showed us is that it's not an irreversible process, that you can reverse the development, right? And so what he did is he took the skin cells from an individual, changed the epigenome by adding chemicals to those skin cells, and made embryonic-like cells. They're called induced pluripotent stem cells. They resemble embryonic cells. And now, over the course of the last 20 years, it's still being defined. We've learned how to take these embryonic stem cells that come from skin or from blood now, and add in new factors and change the epigenome of them again. And then we can derive neurons, we can derive blood, muscles, skin cells, all these different types of cell types. It's really amazing that, 20 years, we've learned to alter the epigenome in this incredibly fabulous way. And so this is just the brief research, and I won't talk too much about it, but this is what I worked on for about 13 years, where I had a collection of 221 people, and I took their skin cells and I made these induced pluripotent stem cells, and then I made heart cells from them, pancreatic stem cells from them. I had these embryonic-like stem cells, and I studied them at a molecular level with the goal of understanding how the epigenome is interacting with the genome at a very detailed molecular level. That was the goal of my project. But to come back to some less detailed scientific stuff and more like, well, what does any of this interesting stuff have to do with me? How's it going to impact my life? And so the induced pluripotent stem cells, one of their key uses is regenerative medicine. And I'm sure everyone's heard about regenerative medicine, but what is regenerative medicine? And that is where we take the cells from an individual and we program them, we reprogram them into this embryonic state. And then we can regenerate. You need a new liver, we'll make you a liver. You need brain cells, we'll make you brain cells. And this sounds futuristic, and it is, but how futuristic, it's up in the air. So right now in Japan, they just recently, I believe it was in March of this year, they've recently approved using this type of regenerative medicine for people with Parkinson's. So they'll take the cells from somebody with Parkinson's, they'll reprogram into them to this very early state, and then they'll make dopaminergic cells. And they'll inject those cells back into the individual's brain. So this is this is in the early stages, but this is the Japanese equivalent of an FDA-approved drug at this point in time. You can make cells that are in the eye, and in people that have glaucoma, one of the problems is the the pressure of the eye is off. And if you drive these types of cells and inject them into the eyes, those cells can restore the correct water pressure, resulting in a decrease in the amount of glaucoma. And this is also in the work for approval for actual medicine. So what do people think? How do people think that this is going to impact us? What do you think the next uses of these iPSC-derived cells will be in medicine?
Oh. All right, she's throwing it open to all of us.
What do you think we'll make?
What will we do with these advances? These
I mean, one
scientific
of the things
advances.
is people that have diabetes, juvenile diabetes, the beta cells in their pancreases they die for whatever reason. So people have been trying hard, and they're coming up with ways to generate pancreatic beta cells. We'll inject these into the correct location of the pancreas. Will completely cure type 1 diabetes eventually.
So that would be a very specific question for us. And if you have other comments or questions in regard to this whole idea of evolving the human body, we're opening it up now to roundtable conversation. And I'm going to be able to be with you for the next 5 minutes. Then I'm going to pass it on to David Sanders, who's going to finish you up as a whole conversation circle. So, David.
Yeah, I was very interested in when you were talking about the microbiome, and I'm curious about that relationship of the care of the microbiome and then the effects on the genome. And
Well, I mean, so if you're eating like high fat food, right? Your microbiome shifts. I mean, the microbiome is composed of, 100 different types of bacteria. And some bacteria do this and some bacteria do this. If you insult the bacteria if you insult your gut, or if you eat a lot of high fat food, the bacteria that you need to help digest that fat grow in quantity, right? And we all need fat in our diet, but by taking in and taking in different types of nutrition through our cells, through our intestine. Well, we can think of it this way. Then if we have this huge amount of this one type of bacteria, and then we eat an orange, right? Is the bacteria that we need to help digest that food and transport that vitamin C across the gut. Do we have enough of it, right? So we can, by altering the microbiome in how we eat and how we exercise it does affect our epigenome, but in an indirect way, because it's affecting how we can adjust or metabolize our food, how we digest our food, what food can we actually utilize.
Renee, you're still here. Did you want to ask any questions before you turn it over to me to then call upon Robert and then Antonio?
Thank you so much for asking. Actually, what I think I would like to do is tell you before I leave about next week, and then maybe you could reiterate it. Next week is Awakening the Mind, and I've decided to take that topic on myself. Maybe David will moderate, and I'll be the presenter. And I'd like to do it from the standpoint of mindful movement for awakening the mind. So that'll be next week's. And I think, Kelly, what a very clear and informative presentation that you've given to all of us. Great springboard for all of you to have a wonderful conversation. And thank you, David, for taking my place from here on out. Thank you so much. Aloha.
Thank you, Renee. And before I go too far, apparently, you were asking about MBSR, and I just wanted to put into the link for the book Mindsight by Dan Siegel, who has done a lot of work on the physiological effects of meditation. And so I put that link there. Somebody says it's not working, so I'll work on that. And at this point, I'll turn it over to Robert.
I wondered, like at the Still and Moving Center, you have a lot of different kinds of movement. And one of the things that's touted these days, particularly, is Tai Chi to balance your body. Does it really make a lot of difference what kind of movement one is engaged in, epigenetically, that is?
I don't think I don't know the answer to that question, right? But I could imagine that if it's like, so I don't know the answer, but we can think about it together, right? So Tai Chi, I think you're actually using your mind a lot to, make the forms that you're making, and it might almost be considered, a meditation type of movement. So I could imagine that could be affecting your body differently than high aerobic, right? Workout. But I think they're all probably, they're all probably healthy for you. But I don't know, like, if you're trying to impact your epigenome of your mind versus your muscles, or I don't know how the differences work.
Oh, well, thank you for everything. It's really wonderful. I just wonder if I could throw in another question. If you eat a lot of sugar, does it make you want more sugar? Does it cause a epigenetic phenomena that makes you
Well,
want
I think that's actually so I don't have much scientific evidence in this, but I think that, our cravings that we have, like some people crave McDonald's and people go, how can anybody crave McDonald's? But honestly, if that's where you're getting your fat and your cholesterol and your protein for, and that's what you've trained your brain, this is where I get these nutrients, then you crave it. right? So your brain craves, when your brain craves something, it's when you're craving something, it's just not that you want to have a bowl of ice cream. It's that your body's telling you you need something in that ice cream. And so if you eat a lot of sugar and your microbiome is set up to digest that sugar, then yes, you will crave it because, you know, even the microbiome, most likely, I don't think that this has been scientifically shown yet, but the microbiome can probably also communicate with your through chemical interactions, communicate with your brain and say, okay, we're set up, we're ready to go. You had a bowl of sugar this morning and now we're hungry. Come on.
Thank you.
Yeah. Antonio, you have a question.
Yes, hi. Thank you. I enjoyed the topic. That was interesting. I kind of always had a feeling about certain things and never like saw the research behind it. So it was interesting to see that that you can alter gene expression, I guess you could say. And but the one that was the reversing of the cell was actually quite still mind-blowing to me. And that's
It's
kind of how I,
still, to be honest, it's still mind blowing to me, and I've been doing it for 14 years now. Yes, it is. I mean, you know, you take a skin cell and you make it like an embryonic cell. It's revolutionary.
I and I feel like that's part of the way that acupuncture works by kind of like the studies have shown that acupuncture puts the body into like a parasympathetic state and that state being in that parasympathetic state helps the body to self-regulate and repair and or from a genetic standpoint puts the proper biological markers in there for proper gene expression, healthy gene expression as opposed to disease gene expression. I was wondering, follow on to some of the other comments, is there a diet that, because like you said, your mind and your gut biome will adapt to the diet that you eat, but is there a diet or have they not done research on that yet? I would suppose as well that is more effective for something like this.
Well, I think there's a lot of different diets, and I'm not a dietitian, but uh, it is well known that what you eat impacts your microbiome, and you know, they don't actually know why the diversity of the microbiome is decreasing in modern society. It could be our antibiotics. It could be that we eat, I mean, we all think we eat these really diverse foods, but we really don't. We probably all eat, each of us has a different diet, but within each of our own diets, we're probably not varying it that much, right? And so the diversity of what we eat might be going down quite a bit also. Right? So I don't know. I would say at this point in time, you know, eat as healthy as you can, eat as diverse as you can. I mean, one time I heard a scientist actually say, think of a color palette and make sure that you eat a food of every color every day. That's one way to simply start thinking about how to make your diet more diverse. If like all the food you're eating is white, that, you know, if it's all orange, I mean, you know, you want a diverse colored palette of food. And that's one way to think about it.
Okay, thank you.
Thanks, Antonio. I see Frida has her
Yep.
hand raised.
Hi, can you hear me?
Hi,
We can hear
Frida.
you.
Hi, Kelly. Really enjoyed your talk. I wanted to ask about the pluripotent potent cells.
Yeah.
How long do they last? So if you develop, let's say, the cardiac cells, and let's say you use it to regenerate cardiac tissue. Now, is that something, or are these now new cells long lasting and continue to reproduce as cardiac cells, or would somebody require, subsequent injections of the pluripotent cells?
Well, so there's a couple things. I mean, people have been trying. That was one of the things with heart failure. Heart failure really is like the fibrotic, the fibrous tissue gets really overtakes a lot of the cardiac cells themselves.
Mm-hmm.
Yeah, cardiac cells don't really divide much in the adult, right? And
Mm-hmm.
so you can coax them to divide in the dish. Once they hit the cardiac cells, you can coax them to divide. They don't go on forever. But what they're actually hoping is that by putting the cardiac cells into a heart. Now, this is not an approved medicine at this point. This is all
Right.
experimental in animals.
Sure. Uh-huh.
So once you take cardiac cells and you put them into the heart, it does improve an infarcted heart. That's somebody who's had a heart attack and now is the heart attack. We now can have people survive heart attacks, but what happens is the heart is gone without oxygen, so it becomes it's been infarcted and it becomes hard, and you undergo heart failure years later,
Mm-hmm.
depending on many things. But they're
Right.
hoping that by injecting healthy cardiac cells into an infarcted heart, that they will release chemicals that will then help reprogram the other cells in the neighboring cells in the heart to become younger and more rejuvenant again. And they have shown that it works. They don't know exactly why. So what I just said is all hypothesis why it's working. It
Uh-huh.
does work. And the cells don't keep dividing. They divide
Mm-hmm.
a few times, but
Uh-huh.
what they do is they help that heart healthy again.
Okay, so those cells are long lasting and they're able to continue producing whatever enzymes or hormones that are going to simulate healthy tissue?
Healthy tissue, neighboring cells, healthy tissue.
Ah, okay, gotcha. Is that the same for the pancreatic cells in the
The
diabetes case?
So for the beta cells are like this holy grail. It's really easy to make embryonic pancreatic tissue. Um, those beta cells are actually quite tricky to make. And so they haven't, unlike cardiac cells, which are pretty easy to make,
Mm-hmm.
it's hard to take a cell all the way to an insulin producing stage. You can do it, but it's hard to do it a lot.
Uh-huh.
So they haven't been doing the same things, but the goal would be that once you make those beta cells, that you can put them into the pancreas and that they would stay and
Uh-huh.
and be functioning. Yes.
Gotcha. All right, thank you so much.
Yeah. Thank you, Frida. Marianne.
Um, that was a really interesting question. I was wondering about the mental environment. We are going to talk about mindful movement, but and you were mentioning the book about mindfulness. It just seems as if somebody is very rigid and very intransigent mentally. They don't like. They just don't like things. They don't like the world. They have a negativity bias going on. Does that impact the viability of these the one types of amino acids that you say open things, or can a mental attitude restrict you and make it more of the methyl type of response where you're just so rigid you don't let your body go through the epigenetic support?
Well, so this answer is outside the realm of what's scientifically known, okay? But I'll answer it anyhow. So, in my opinion, what could be happening is that just like if you're under high stress, you're in a war zone, you're in a life or death situation, that can modify your brain. I mean, really what it is, this is very basic animal modification, right? You know, you're young, you walk to the edge of the cliff, you walk off the cliff, somebody grabs you and you're like, don't do that, right? Your brain gets, okay, somehow that memory is formed, somehow the memories are formed through epigenetic modifications. So if we start being stiff and our mind is stiff, and that is modifying our epigenome most likely, right? And so, you know, forcing laughter on ourselves. I mean, they say laughter is medicine. It's probably, it's the chemicals that our body produces that interacts with our brain impacts our epigenome and controls the DNA, which then again releases what goes into our body, tells our body what to do. So I would say that more than likely, people that are always stressed, people that are always happy. I mean, I don't think you want to be on either extreme. Those are, you know, manic extremes. But their epigenome are probably set up. They are setting up their epigenomes to be like that. You know, I think vitamin D, it affects the body, but also the mood. And it really is a very potent epigenetic modifier. And vitamin D is impacting the epigenome of our brains, and it impacts our moods. And that's a scientific fact. Much of what I said is not, but that's a clear scientific fact.
Mm-hmm. Kelly, have you run across the work of the book called The Biology of Belief?
No.
Biologist. Somebody know his name right offhand? Fascinating book on the topic of, he was the first biologist who was able to extract the nucleus of a cell and to discover that the function of the cell was not, in fact, based upon the nucleus of the cell, which biology had previously believed, but was in fact the function of the cell had more to do with what the receptors on the cell itself were sensing from outside the cell.
Oh, yes. I don't know that book, but yes,
Bruce Lipton.
Yes, that is very true. I mean, we have our cell, the cell interacts with the rest of the body through receptors. And I'll come back to vitamin D. You have a vitamin D receptor, right? The vitamin D itself doesn't enter the body. The vitamin D binds to a receptor, and that releases the receptor from the cell membrane, and then that receptor goes into the nucleus and alters the DNA in the epigenome. So I don't know that particular scientist or that book, but that is a concept I'm very familiar with. Yes.
Okay. It kind of ties these ideas together that how we think and our emotions ultimately affect the body and its functioning.
That's very true. Yes, it's very true. And so trying to be positive, trying to, and it's a two-way street.
Mm-hmm.
What we eat, what we do affects our brain, how we think, what we watch, right? The TV we watch, the movies we watch, the whatever we read.
I see something in the chat here. Okay, all right. Okay. Did anyone else have a question burning right now? If not, I'm just curious. I can imagine you said growing a liver. I'm thinking of a chunk of liver. How does that get delivered into the body? Or am I missing?
Well, they haven't done that yet, but actually the liver is one of the few organs in the human body that can regenerate. So you can actually remove, I know at least half, maybe up to 2/3 of the liver from somebody, and it will regenerate itself.
Mhm.
And so, why would you want to do this? Well, sometimes people have a drinking disorder and the liver gets destroyed. And so if you could regenerate a liver that you could give them. Now, they are also thinking of using like pigs and liver transplants happen not infrequently, right?
Mhm.
And so they take a liver from somebody who's recently passed and we'll put it in somebody who needs a new liver. But there's all types of issues with that because our body recognizes ourselves and then tries to reject anything that's not ourselves. It's one of our immune functions, right? But if you could take my skin cells and turn them into a liver via induced pluripotent stem cells and grow a liver in a dish and put that back in, put healthy liver cells back in, our body would not reject it.
Mhm.
Our body would see it as self. So it would get around all of these issues of intolerance from exploitation.
Mhm. Yeah. I see a question in the chat. If someone is diagnosed with ADHD or some kind of neuropathic condition, can you reverse the conditions by lifestyle changes?
Again, I don't know. I'm not a physician. I know that ADHD, there's a large spectrum of it. I think that some aspects you probably can help, right? So we'll go that way.
Mhm.
You know, what you how you want to interact. That's a touchy topic because I don't want to say yes, you can do things and it will go away because that's clearly probably not 100%. But you know, touch and food and interacting with people and social context. I'm sure that the epigenome of somebody that has ADHD does not work differently than neurotypical people. So yes, you can modify the epigenome of somebody with ADHD just like you can a neurotypical person.
A good book on the topic of ADHD and its genesis, Gabor Mate, MD, and a book called The Myth of Normal might be a good way to research that. I'm noticing the time. It's 4 o'clock on the nose. Kelly, are you right across the street, Ali'i? Or
I'm a few more down in Vaea.
okay, gotcha. All right. So I'm wondering, is there someone on board that is feeling the compulsion to offer a vote of thanks for our guest speaker today? Anyone jumping into that? In which case, I'll do it. Kelly, thank you so much for sharing your wisdom with us and intriguing us and maybe leaving us with even more questions than we had when we began, which is, I think, a really great sign for our human minds, which are constantly yearning for a challenge to grow and expand. So thank you very, very much on behalf of everyone here. And we all look forward to seeing you all next week as well.
All right. Thank you. Bye-bye.
Thank you,
Kelly.
Kelly. Great talk.
Thank you. Thank you, Kelly. Happy Easter,
Thank
everybody.
you.
Oh, happy
Happy
Easter.
Mary.
Yeah. Thank you, Mary.